Understanding the molecular evolution of SARS-CoV-2 is key to developing effective antiviral medicines and vaccines to end the COVID-19 pandemic. By counting the total percent of mutations occurring at each site over the entire SARS-CoV-2 genome, using over 11,000 selected SARS-CoV-2 genomes from infected human patients, some gene regions, like the spike gene in brown or the Nucleocapsid gene in yellow, exhibited high mutation rates, or peaks, which may increase the virus's fitness, like in the yellow or positive selection below. Most other gene regions, like ORF1ab in green or the membrane gene in purple, exhibit no mutation rates and maybe cannot tolerate mutations, shown as negative selection in red. They may also exhibit lower mutation rates than the high substitution or mutation rates, which are random and may not affect the virus fitness at all, that being neutral in green or near neutral selection in light green. Converting this, the probability distribution of a particular site undergoing 0 to 1 to 2 mutations or even more exhibited an L shape. This means that most sites exhibited little to no mutations and are under negative selection or are strongly conserved. Sites with increasing mutation rates relative to the genomic mutation rate are exceedingly rare. Knowing this, which theory can best explain this L-shaped distribution of the relative mutation rates for SARS-CoV-2? Selectionist theory states that most observed mutations are under positive selection in yellow. Neutral theory, KNT, states that most observed mutations are under neutral selection in green. Near neutral theory, ONNT, states that most observed mutations are under near neutral selection, light green, with an asymmetric distribution of weak negative selection, which actually slows down the mutation rate. None of these theories are able to explain the L distribution except for strong negative selection part in red. Selectionist theory, ST, overestimates positive selection but underestimates near neutral selection. In contrast, KNT overestimates the true neutral selection, yet underestimates the positive selection. ONNT underestimates the near neutral selection of slightly positive mutations. Therefore, we propose a near neutral balanced selection theory, NNBST, as a hybrid theory of these two kinds of ST and KNT theories, comprising both of their critical factors: positive selection and near neutral random selection, encompassing both weak negative and weak positive selection. The NNBST theory states that most sites are conserved, as in agreement with the previous three theories. Some sites are under near neutral selection as a symmetric distribution of both weak positive and weak negative selection, and very few sites are under positive selection. If you are interested in this work and like to know more, look in the link below in our description to our full-length video. We also have a comparison of the SARS-CoV-2 mutation rate at the temporal level over time, again showing that these first three theories do not adequately explain the mutation rate for this virus, however, is instead consistent with NNBST.